Hi, Michelle Glass here. Welcome to our last episode in Chapter 9, Part 1, looking at Mendelian genetics and patterns of inheritance. With Mendelian genetics, you're looking at characteristics that are coded for by a single gene, and there are some of these that are true for humans. We'll be finding in Part 2 that inheritance is often more complicated than the Mendelian pattern of inheritance. With human genetics, there are several things that are inherited as single genes. This is not an entire list. This is just some. Some of these are considered genetic disorders that can cause medical issues for people, such as cystic fibrosis or sickle cell disease. Some of these are just variations that exist in the human population, although typically at low levels, such as albinism and achondroplasia, which is dwarfism. Some of these are just physical features that we've described before, like thumb shape, either hitchhikers or straight thumb, hairline straight or widow's peak, freckles, that really have no serious impact on your ability to survive and reproduce. Characteristics that are called dominant traits are not necessarily named this because they are the most prominent in the population. The dominant trait doesn't mean that it is the normal or typical form. It just means that it is controlled by the dominant allele, meaning that if you're homozygous dominant or heterozygous, you would have the dominant form. Freckling is a good example of this. The recessive form of this gene is actually no freckles, and that is the most common in the population. To have freckles is actually rare, even though it is controlled by the dominant form. Now, if you have a lot of freckles and you cross with somebody with no freckles, you might see either 100% of your offspring being freckled, or you might see 50% of your offspring being freckled in the case of a heterozygous crossed with a homozygous recessive. How is lack of freckles more common? Well, if that is the most common form in the population, then you're getting homozygous recessive times homozygous recessive, and all of the offspring would be then homozygous recessive. Again, it's important to pay attention that dominant traits are not the most prominent in the population. We see that in some cases they're rare, such as polydactyly, which is the presence of maybe a sixth finger or toe or webbing between fingers and toes. Achondroplasia, as mentioned, dwarfism is also a dominant trait, but rare in the population. Huntington's disease is another example here. Some traits are recessive traits, and these are often what we see for our genetic disorders, because very often individuals with the recessive form are not able to survive to reproduce and pass on the recessive form to their offspring. The significance of recessive traits is that they can be passed on by individuals who are phenotypically normal or unaffected. We call those individuals carriers. So remember, to have the recessive trait, you have to be homozygous recessive. So a person who is heterozygous does not have the characteristic, but does have that allele, which can be passed on to offspring. So we can see if you have two carriers crossing, a small percentage of the offspring, 25% probability that you're going to see that recessive form showing up. Now, individuals who know that they have a family history of recessive traits in their ancestors, they might consider genetic testing to see if there's any problem as they are facing possible parenthood. So maybe either before choosing to become pregnant or during the pregnancy. There are a few fetal tests that can be done. These first couple are invasive, and they do have a 1% risk of problems or termination of the pregnancy, so they're not done unless there is a reason. Amniocentesis is the first described here. This can happen weeks 14 to 16 during pregnancy. The needle has to go through the mother's abdomen into the uterus to remove amniotic fluid. Then you have to culture some of those fetal cells to look for chromosomal abnormalities or conduct some biochemical tests to look to see if there's problems that can be detected that way. Chorionic villus sampling is a faster test. It can happen earlier in pregnancy, 10 to 12 weeks instead of 14 to 16 weeks, and you get your results back within 24 hours. So this is often preferred. In this case, you are taking the needle through the vagina and cervix in order to remove cells from the placenta, and then you right there have those fetal cells that you're able to use for the genetic analysis. A third type of testing is the ultrasound. This is not invasive, and so this is typically safer and more routine part of your prenatal care. Ultrasound can occur around 20th week of your pregnancy. This allows you to get a visual image of that developing fetus, and so sometimes you're able to detect possible abnormalities there and then do further testing as needed. Okay, so that is it for part 1.